Hi, everyone. We're going to kick off our seminar. Thanks for joining us. This is the June 2025 seminar on physical genomics. My name is Benjamin Keene. I'm here at the Center for Physical Genomics and Engineering at Northwestern University. We're excited to welcome our invited speaker today, Professor Dawn Chen. She is an assistant professor in the Biomedical Engineering Department here at Northwestern. Her lab develops next generation optical imaging modalities and computational algorithms to study a wide range of living systems, including cell cultures, organoids, tissues, and live animals. Prior to joining Northwestern, Dr. Chen was an NIH K99 awardee at Cornell University, and she completed her postdoctoral training at the University of Illinois in Dr. Gabriel Popescu's Quantitative Light Imaging Lab. Dr. Chen earned her PhD in 2019 from University of Miami. Professor, you'll be telling us a lot more about your research. Thank you so much for joining us today and sharing your work. One quick note for our audience. We have a Q&A session at the end of the talk. So you can type any questions that come to mind into the Zoom chat when you think of them, and we'll address them at the end. Or you can just wait till the end and chime in then. If you'd like to learn more about the research and training programs we have here at the Center for Physical Genomics, you can visit us at physicalgenomics.northwestern.edu, or you can check out our online archive. We've got recordings of all kinds of talks, seminars, symposia on our YouTube channel at PhysGen Center, or you can follow us on Twitter X or Blue Sky. So at this point, I think I can turn the proceedings over to Dr. Chen, and we'll get started. Thank you once again for sharing your work.
Thank you so much, Ben, for the introduction and invitation. And hello, everybody. And it's really nice to meet everybody here virtually on Zoom. And for this talk, I'm going to focus on some of our efforts to develop systems for like a variety of biological systems, including the organoids, brains, and the cells. So let me give you an overall picture what I'm going to talk about today. So today we're going to focus on the imaging systems. Now we still want those imaging systems to have the subcellular resolution. So going to be like 0.1 micron to 1 micron resolution-ish. And for the imaging depth, we want to image a variety of different biological systems. So we will have single layer cells that will give us like about imaging depth about tens of microns, but we still want to keep the resolution to be diffraction limited. So we use a visible light, so about 300 nanometers. And another thing is that we want to keep this phase sensitivity. I'll talk more into what is a phase, I mean by phase imaging. So for the phase sensitivity, we still want to keep it as a nanometer sensitivity, so we can measure really small changes in the nanoscale. And we also want to develop systems that are suitable for organoid spheroids imaging, which gained a lot of interest compared to the 2D single layer cells for like drug discovery and the other applications. So for those type of image organisms, they are about hundreds of microns to a millimeter. So they're like a spherical shaped. So we need to have an imaging system that is able to image like deeper into them and have better sectioning to look at each layer of the cells inside of them. And we also want to image the thick tissues and of course the in vivo animal models. So those are more challenging because now we need to have an in vivo system inside of live animals, and we need to be able to see through this whole millimeter depth of tissue. So we'll also go a little bit on that. And so we want to develop multimodal imaging for living organisms. So why is that? So multimodal imaging, for example, quantitative phase, fluorescence, and harmonic generation. So different imaging modality will give us different contrasts. And it also offers us different measurements. So we can look at different targets in our environments. For example, for the quantitative phase, we can look at the morphology of the cells. So we can those morphology information will give us a certain specificity or variability information. And we can also retrieve the strain mass information that comes with the quantitative phase imaging. And using those strain mass information, we can look at the cell growth or death events. And we can also further retrieve the refractive index of the cells. And using RI, the refractive index, we can look at the cellular dynamics. And we can also, using the phase imaging, to look at the fluctuation of the membrane of the cells and to look at the stiffness of the cells. And we, of course, love fluorescence, and fluorescence is widely adopted in most biological, like almost all the biological labs. So using fluorescence, we can, first of all, most important, we can look at the molecular distribution. It gives us the biochemical specificity. And we can also use the fluorescence indicator to look at the calcium or voltage signal, to look at the neuronal activity or the cell functions. We can look at the lifetime of the fluorophores and to indicate the metabolic activity of the cells. People, of course, also doing the spectral, looking at the spectrum of the fluorescence or the shift of the spectrum of the fluorescence to look at the protein interaction and the mobility. And the last modality that I want to include in our imaging system is harmonic generation. So for the SHG, the second harmonic generation, it mainly comes from the non-central symmetric structures. And using SHG, people mainly focus on looking at the collagen fibers or microtubules. And for the THG, the third harmonic generation, the signal comes from the refractive index mismatches. So we can look at the lipid membranes or organoid boundaries. And on top of that, we will develop, like people have been developing computational algorithms to enhance the imaging ability for each different modality. So let's look at the single layer cell, the easiest biological organism first. So in this slide, I want to introduce what I mean by phase. What is quantitative phase imaging? What is this phase that we're measuring? So for most thin biological samples, we can consider them as an amplitude object, meaning you have an incident wave, incident light, and when it enters this sample or scattered by the sample, this sample changes the amplitude instead of the amplitude of this light electric field. So what you're seeing on this picture, can everybody see my mouse cursor? Or should I use another different laser pointer?
I can see your mouse.
Okay, now probably clearer. So you can see that for amplitude object, you have a wave coming in, and then it changes the amplitude of this wave. And for phase object, instead, for most thin biological samples, they are not amplitude object, meaning they do not change the amplitude of this electric field. Instead, they delay or advance this electric field. So you have the same incident wave in, and this is your phase object. So it delay or advance this wave by a little bit. So compared to the incident wave, after the scatter wave coming out, you will have a phase delay, this delta phi over here. And this is the target that we are measuring in quantitative phase imaging. So you can think of a very simple cartoon that you have a plane wave coming in, and this is your cell. And so based on the different thickness and the refractive index difference, you will have different delay at each point in the cell. So if you estimate using geometrical optics, very easy, you can approximate this phi as 2 pi d, the refractive index difference between the cell and the media, and of course your imaging wavelength lambda. And the straw mass quantity is linear proportional to this phi. And this gamma has been studied a lot by previous researchers, and this is just a constant. And of course, it depends on the biological samples, which type that you're imaging. And for well, we have before the quantitative phase QPI developed, people have been developed enhanced contrast enhanced techniques For example, the DIC, the diffraction interference microscopy contrast, DIC differential interference contrast, or the phase contrast microscopy. So those techniques are aiming to enhance the contrast in this thin phase object biological samples. However, using those techniques, you do still have a background, this large background in the picture that limits the contrast in your image. And the one reason for QPI to develop is that if we use a QPI technique, we can further enhance the contrast by removing this DC background. So you only have left with this delta I signal in your QPI image, so we can further enhance the contrast. So this is a good example to show you the difference between a phase contrast versus a QPI method. This is imaging of a microtubule. So in the phase contrast image, you can barely see this microtubule structure. But after you remove that background, if you retrieve the phase information, all this electric waves, electric field, then you can see the microtubule structure. So QPI refers to a large group of methods. And today we're only going to focus on the methods that are using an interferometer. And the reason to include an interferometer is that having this, it will enhance your contrast even further compared to the QPI method that doesn't use an interferometer. And we also want to keep it as common path because if you have a common path QPI system, you will enhance the stability and sensitivity. Remember in the first slide that I mentioned for all the imaging modality I'm talking about here, we are going to have a nanoscale sensitivity, phase measurement sensitivity. And we also want to focus on the system to have a broadband illumination because broadband illumination will give us the sectioning ability. And also for even the in vivo model imaging, we want to even further enhance the sectioning ability by introducing a pinhole or harmonic generation. So when we use QPI imaging to image any object, what we get is a bunch of images. And you can think of those images as a distorted version of this object. And the end goal for all the imaging systems is to try to get the best estimate of what the object is. And for the scattering-based imaging systems, the end goal is to solve this inverse scattering problem. And that is some quantity that we call the refractive index, the cells. And the refractive index are the intrinsic properties of the cells. So those is like a mark on the cell that doesn't change, that we want to retrieve those information so we can really look at what the object is like. And we developed a method called Wolf phase tomography. So the benefit of using this method is that this method considers the broadband illumination properties of the light. So for the previous methods, they do not consider this broadband. And because we say that we want to focus on the QPI that has a broadband illumination, so you really need to take into account the property of the broadband source. So this is something we call the gamma. So this is a correlation function. So this function is basically an ensemble average of the multiplication of two electric fields. So PQ here can be either I or S. I denotes the incident, and S is a scatter light. So the correlation function is an ensemble average of a product of U incident light or scatter light. So it can be II or IS. So it's correlation function of the autocorrelation of the light or the correlation between the incident and the scatter light. So if we get those information, this gamma term, and then we can retrieve this using the WPT algorithm, we can retrieve the refractive index of the object. And this quantity gamma here is a complex number. That's why we do need to know the phase of the electric field, which we already have that information from the QPI system to solve this inverse scattering problem. So for the gamma I term, it's very easy to retrieve. We simply need to know the spectrum of the light. And then by doing the Fourier transform, we can get the autocorrelation of the light, and then we can get the second derivative of the light. And for the gamma IF, that term is a little bit trickier to solve, and then we need to depend on imaging modality that can give us the QPI, the phase term. So what we did in this project is that we use a phase contrast microscope, and we have a SLIM add-on. So SLIM stands for spatial light interference microscopy. It's one of the techniques that we developed earlier in Dr. Gabby Papascu's lab at UC. So it's basically phase shifting interferometer, common path interferometer. So using this modality, we can retrieve this gamma term, which is a correlation between the incident and scatter light. And with that, you have a set of images, and you can retrieve the refractive index of the object. For example, so before we move on to the biological sample, we test on some known refractive index sample. For example, this bead. We know the refractive index, and now we can validate this method using this known object. And after we validate this method, we can move on to the other samples. So we can see that the refractive index does give us a certain level of specificity to tell you where is the centriole of the sperm cell, where is the nucleus, microsome, and also the axial filament, because they have different refractive index of those cellular compartments. And because this is a label-free method, so you really need to, you really only need a minimal level of power to image this living cells. So we keep the light below 5 milliwatts, and we image over 5 days. The cells are happy, and you can just image long-term and do your longitudinal studies on the live cells. And the refractive index, another benefit is that using this quantitative refractive index mark, you can see the cell growth, cell death. You can also measure the growth rate and other properties of the cells over a course of like a long period of time. So second topic, let's move on to a more challenging biological objects, the organoids and spheroids. So when we image a thick sample, the difficulty is that, so remember what we said about a little bit about coherent scaling, which we use a broadband illumination to get rid of the out of focus light. So only within this coherence volume, you can have a meaningful signal from your imaging modality. However, using the coherent scaling, you can see that we still have a limited ability of rejecting the out of focus light. You do still have a pretty poor optical sectioning when you focus on the Z that you don't have any object. So there's another way to further enhance this optical sectioning ability is to use a physical pinhole to reject the out of focus light. And confocal, which is very popular among a lot of biological labs, this is to the most popular way to image in a spheroid, to use a confocal. So, however, confocal, usually you need to fix them, stain them, and usually cells are not happy, they're dead. And you can use a live imaging dyes, however, you still have a different, limited ability to distinguish each spectrum. You have a limited emission bandwidth to look at the different cellular cellular compartments of interest. So if we can do a label-free imaging of this spheroid, and then we can use a computational algorithm to retrieve those biochemical specificity, then that will be the best, because then we can do label-free, no staining, and we can image with minimal power, and we can image over a long period of time. So now, because of the AI development, the machine learning development, this problem can be solved. We can use deep learning to convert the images images on the left to the images to the right. So this is the system that we developed to do this task. So it's basically a multimodal system, including a QPI method and a confocal fluorescence imaging. So for the QPI part, we adopt a method called DIC, the differential interference contrast, plus common path interferometer that further introduced by the LCVR. So LCVR stands for liquid crystal variable retarder, which will give you the phase shift between the two polarized light. And then using the phase shift algorithm, you can further retrieve the phase of the object. So now using this multimodal system, we can have a colocalized pair of QPI images and confocal fluorescence images. And now we use those pairs of images to train the network. So after the network is trained, the network will tell you, after you input label-free QPI images, the network will tell you the fluorescence corresponding images that you trained on. And this is a graph to show you we do still have a nanoscale spatial and temporal sensitivity. So LS-GLIM stands for the laser scanning gradient light interference microscopy that in this project, we developed in this project. And the GLIM is a previously developed in Gabby's lab, which is a wide field version of the GLIM, laser scanning GLIM. So we can see that the laser scanning GLIM has a better spatial sensitivity and also has a better temporal sensitivity. And both of them are have the nanoscale sensitivity. So before we move on to the unknown sample, of course, we test the known sample, for example, a bead. So we can see that using the ACM method, you do, when you input a label-free image of bead, it does give you back the fluorescence imaging of a bead, and it also enhance axial resolution and sectioning. So now we can move on to the unknown sample. So for QPI, another thing is that we can use those QPI information to retrieve the dry mass information. So this is a dry mass information that retrieved by the QPI and the fluorescence images that generated by the ACM method. So we can look at the volume of the cell, and we can also look at the dry mass density of the cell. And for this project, we also trained the network on two different fluorescence signal, which is tau and the MAP2 of the neuron. So the network is able to generate both two channel of computed fluorescence signal. And using those two signal, we will be able to tell where is axon versus the dendrites. And we, of course, the main aim of developing this is to imaging organoids. So we can see that after you input the label-free phase images into the network will output computed fluorescence, confocal fluorescence images that look like the ground truth, which is provided by staining and imaged by the physical confocal fluorescence microscopy. And we can further, we further developed automatic segmentation of the cells inside of the spheroids. So we can segment those cells and we can count the cells and we can look at the distribution of those cells in terms of dry mass, volume, and different, like the volume, dry mass of different compartments in the cells of interest. And the network that we trained on were using the spheroids in PBS, and those are not it's in the pancake shaped. It's not very spherical, but when you put the spheroid in the hydrogel, they will tend to grow into a more spherical shape. And we never trained the network on the samples that grow in the hydrogel. However, our network can still be able to pick up this spherical shape of the spheroid that grow in the hydrogel. And based on this imaging modality system, we also developed a health variability assay for mouse embryos. So now we don't need to stain or doing fix the sample. We just retrieve the embryos, keep them alive, and then you can image them over days. And then you can, the network, the computational algorithm will tell you whether this embryo is a sick or intermediate or healthy sample based on this morphology information of the label-free QPI images, and also we retract the features using those images, and also using the ACM-based method, we can also look at the fluorescence signal that computed by this network. And overall, have all those information included, we can the network will give you a score to indicate the healthy level of this candidate. So lastly, let's move on to a even more challenging system, which is in vivo animal models. Now we have a live animal, so we need to we have a lot more like issues involved with the live animals. For example, the motion artifacts, and also we have all sorts of cells inside of this animal that we need to image through. So for the in vivo systems that the previously introduced method were all based on the transmission mode, which are not suitable for the in vivo system imaging, because the animal in vivo imaging, you do need to have a reflective system to image through this whole animal of structure of interest in the animal. So what we did is that we developed scattering-enabled API quantitative phase imaging. So API just means reflective. The source and the detection are on the same side. So using this, we'll be able to image. This is very suitable for the in vivo imaging. And as I said, because we need to further enhance the sectioning ability of this modality, so we introduced a confocal pinhole to the phase to the DIC system, and we still adopted a phase shifting common path interferometer as we did before. And we also want to have the specificity, and we eventually want to image calcium signal of the neuronal activity. So we introduce a three-photon fluorescence microscopy to do that. And we also include the third harmonic generation to look at those membrane membrane on the lipids and the overall cells. And for this API QPI system, because we introduce a confocal pinhole, so to retrieve the phase, actually, we have two different scenarios. The first scenario is that for the 2D sample, when the light comes in, you do have this incident light that is reflected by the substrate. For example, you have a cover glass over here, and that cover glass will reflect the light back towards the detector. And with this 2D, the incident light referenced samples, you have a different way of constructing the phase, which is the same algorithm as what we did before, because for previously, when you have a transmission systems, you always have this incident light coming through the detector. So you have the same way of constructing this phase using the previously introduced algorithms. However, for the 3D samples, when we images 3D like organoids or brain, because of the pinhole, we actually reject all the incident light. So all you're detecting is this backscattered light that doesn't have any incident light information. So here you only have this US, and because of DIC microscope, you have another closely shifted US, another light that coming in. So you have another US. However, you do not have this UI. This UI is important, however, in the QPI algorithm, because UI serves as a reference to tell you the relative shift between the UI and US. And for the 3D samples, now you do not have US anymore. Sorry, you do not have UI anymore. You do not have this reference anymore. So we need to introduce a new reference. So now we introduce the summation of the two US and US prime as reference. This U, the summation of two complex variables. And this U now serves as a reference. This is a very key it seems trivial, but actually it's a very key step to do in this algorithm. If you do not introduce this hue, if you only calculate the angle between this US and US prime, you will have a lot of problems. Like because you don't have a reference field, now you have a lot of phase wrapping problem, and you will lose the meaningful information of the biological samples. So this is what we kind of developed for the 3D, we call the incident, we call the self-referenced scenario. So we introduce a U to have a new reference that we can have the meaningful phase that introduced by the sample. So before we move on to the unknown sample, we of course always test this method using the sample that we do know the phase shift. So this is a 2D case, which is a mix of 1 micron and 3 micron beads in the oil and on the cover glass. This is actually on the mirror. And we can test whether this phase shift that we measured using the CQBI agrees with the theoretical shift that is introduced by the sample. That they agree with the theoretical values very well, and you can see that the phase shift of the 3 micron samples is exactly 3 times of the 1 micron samples, which is what we expected. And we also need to test the algorithm that we developed for the 3D samples, so which is a three-dimensional. So we use the microbeads. Those are all a mixture of 1 micron non-fluorescence and fluorescence beads in agarose, and then we image through a whole thick slab of 700 microns using CQPI, and we can look at the phase shift that introduced by this 1 micron in the agarose, and they also agree with the theoretical value. So now we validate this CQPI method. We can use it for the samples that we do not know the phase shift. So this is again an organoids. Because of this organoids grow in the PBS, they're more like the morphology is like a pancake shaped. So on the top left, what you're looking at is the nucleus of the organoids that is imaged by the three-photon microscopy. And this column is a third harmonic generation for signal coming from the organoids. And this column is for the CQPI image of the organoids. And the last one is the overlay of all the signals. And when we zoom in to the individual cells, you can look at, so this red channel is a 3-photon channel, the green channel is a THG, and the gray level is a CQPI, and then the last column is an overlay. We can look at the single cell morphology using this CQPI, and we can also see the nucleus of the cells in the CQPI. So now we move on to like more challenging biological system, the in vivo mouse brain. This is a wild type mouse with a virus injection, and we create a cranial window to remove the skull, and we are able to image this in vivo mouse brain up to about 1.1 millimeter using both like 3 different modalities. So now we are looking at the white matter. So this is just a 3D reconstruction, the rendering of the images, the 2D images that you just saw. So for the fluorescence signal, the virus injection targets the nucleus of a certain group of inhibitory neurons. So it's not all the neurons, it's some of the inhibitory neurons in the mouse brain. So we can see that all the nucleus up to a little bit in the white matter region, because there are not many cell bodies in the white matter region. And this is a THG signal that will give you some the axons and also the capillary and other the boundary of the cells. And this channel is a CQPI. This big thing over here is a major blood vessel inside of the mouse brain. And then we can also look, all the signals will show in this channel. Basically, we have all type of cells, all type of structure, the axons, the blood vessels, and of course, other type of cells, all showing up in the CQPI channel. And this is the final overlay of all the three channels. So we can look, we can have a closer look at the images from the three channels. So the fluorescence will give you the nucleus of a certain inhibitory neurons inside of the brain, and THG will mainly show you the small blood vessels and axons, and also the cell body is not very good. In the THG, it mainly shows as like a dark circle. Those are the individual cells. And in the CQPI, we can look at all different structures. So this big circle thing is a major blood vessels that go through this imaging slab that we imaged. And we can also look at the axons, the dendrites, and also different individual soma of the cells. And the last column is a confocal DIC images. And the CQPI compared to the CDIC, we can see clearly of the neuron body I will show you more images while we zoom in to this smaller region of the images. So those are still in the neural cortex. And then we can image the white matter region inside of the mouse brain. And it's the CQPI is very similar to the THG. However, it's much faster. I will summarize that in the last slide. And it's very good for looking at the morphology of this overall white matter region. So when we zoom into those images that you just saw, so we can look at the morphology of single neuron. So this is the nucleus again. This is the THG channel. As I mentioned before, in the THG channel, you do not see a very clear boundary of cells. So those are not very good for looking at the individual cells. However, in the CQPI channel, you can actually see the boundary of this soma. And then you can look, you can also kind of see the nucleus inside of this soma. So those are just the images, the corresponding the localized images at different depths. And we were able to find the neurons in the whiter, some of them in the white matter region, about 830 microns. And another benefit of a CQPI is that because the CQPI is a quantitative phase information, it does not depend on the imaging parameters that you're using. For example, if you enhance the light, if you increase the gain of the detector, the quantitative phase information will not change. With that, you can actually have using this information to analyze your biological samples in a meaningful way, in a long-term way. You can look at the dry mass distribution at different time points and look at the dry mass distribution of the structure that you're interested in. So now we're interested in the nucleus. So what you're looking at, this is a Z slice. So if you have a stack of images, if you slice them along the Z, you will have this. So this axis is imaging depth, this imaging Z direction, and this is the horizontal direction in the images. And then you can look at the different structures. So this is a major blood vessel, and you can also find individual neurons that lights up. And when we zoom into the XY plane, we can look at the boundary of the cells, and we can look at the nucleus, colocalized nucleus inside the cells. And interestingly, we actually find that the soma is actually have different shapes. Some of them are like more oval shaped. Some of them are like pentagon shaped. It's quite interesting. I've never seen like the previous imaging modalities that reported this kind of different shapes in the soma of a neuron. And now we have we have the QPI information. We also have this fluorescence information that tells you where are the interests locate inside of the biological sample. We can use those two to combine them to look at the dry mass of the nucleus. So this is the rendering of the dry mass of the nucleus, and the purplish color indicates the dry mass of the white matter. If you want to look at the white matter changes over time, you can look at the dry mass of the white matter. So in summary, the CQPI is based on the linear scattering, and the multiphoton microscopy is based on the nonlinear excitation or the harmonic generation. So because of linear scattering, CQPI does not require a femtosecond laser. So you can just use a CW, very cheap laser, to do this task. And because it's linear scatter, so photon budget in the CQBI is very high. And for multiphoton microscopy, the photon budget is always very low. It's photon starved. And for the tissue damage, because we based on linear scattering, we have a bunch of photons coming back. So the tissue damage is minimal. We only shine light that with minimal power on the sample. However, for the multiphoton image, we need to be more careful. We need to limit the average power to prevent the heating, but we also need enough very high pulse energy to have this 3-photon signal. So when I say very high pulse energy, it's usually about 1 nanojoule for 3-photon case. And we also want to maintain this maximum pulse energy and the peak power to prevent within the safety level to prevent nonlinear damage. And because in the CQPI we have a bunch of photons, so we can image very fast. However, in the multiphoton case, for the structural imaging, it's actually quite slow. For the deeper region in the set of the brain, you need to integrate about 20 seconds to have a good SNR. However, in the CQVI, you can just directly, immediately see the structural information. However, multiphoton image is very good for the calcium imaging, which is used the main use of the multiphoton imaging in the in vivo animal models. For both modalities, we have a resolution about 1 micron, and imaging depth, both of them can do 1 to 2 microns. And this the CQP I have shown is limited on about 1.1 micron. However, we are working on further enhance the imaging depth for CQPI to 1.1 millimeter to 2 millimeter using adaptive optics. And the specificity for the CQPI, because we have the morphology information, so we have a certain level of specificity. However, we do not have the biochemical specificity, and that is introduced. We can introduce by the multiphoton microscopy. And for the cost, the CQPI, the most expensive part is the femtosecond laser, because we do not need that anymore. So the CQPI the cost can be low. And for multiphoton image, it's an expensive modality. So with that, I would like to thank my great mentors, the late Dr. Gabby Pascu, who trained me on the quantitative phase imaging at UIUC, and Mark Anastasio on the computational algorithms and machine learning project, and also the Chris Xu, who trained me on the 3-photon imaging at Cornell. And here are our lab members and collaborators and our sponsors. And we also have available positions in my lab. So this is my lab page, the XDeep Intelligent Quantitative Imaging. And if you're interested, please send me an email. We are hiring. Thank you so much for your attention, and happy to take any questions.
Thank you, Dr. Chen, for a fascinating talk. I think we now can open the floor to any questions, either speaking or texting. Okay, we have a question from Gengwan. I don't know if you want me to read it or if you want to do it. So what is? Go ahead.
Is it in the chat?
It is in the chat, yes. And the question is, what is?
Yeah, I can read the message to all the
Okay.
audiences here. So the question is, what is the dry mass indicator mainly used for? What function of the cell is it related to, or what information of the cell can it reflect? So this is an awesome question. So the dry mass information is basically, remember that the phase that we imaged is proportional to the refractive index difference, right? So it's n of the object minus n0. N0 is the refractive index of the media. For example, if the cells are in the water, then n0 is 1.33, the refractive index of the water. So we are measuring this difference, right? So, and the dry mass, you can think it as a non-aqueous content of the cells. So for example, if you remove all the water from the cells, the rest of the mass is what we call dry mass. So this dry mass is basically contributes from like proteins, the DNA, RNA, and the lipids inside the cells. So those mass of those matters, if you remove out the water, the non-water part of the cells, those will all contribute to the dry mass. And this dry mass indicator is basically tells you the concentration of those different things that I just mentioned, like protein, DNA, RNA. So for example, in the nucleus, you have a lot of DNA, RNA, and also proteins inside of the nucleus. And you have a denser compact like compartments. And then you will see that phase is higher and the refractive index is also higher. So that gives you like a certain level of specificity of the different compartments of the cells. Also, you can use the dry mass information to, as I said, quantitatively analyze in the longitudinally how they changes. For example, if your drug is targeting some parts, let's say let's just say shrink the nucleus of the cells, then you can look at this same cell longitudinally and look at the dry mass density of this nucleus. And then if this drug works, maybe, and then you can see the changes in the dry mass of the nucleus. So this is just one example. You can do all sorts of the region of interest that you're interested in and using dry mass to give you a quantitative measurements. And the next question from Igor is, have you imaged, hello, have you imaged muscle or heart tissue? What depths would you expect to achieve? So so far, I have not imaged muscle. Actually, we did image one muscle, the muscle on the mouse leg. We did image once. And I think that was using the visible light modality, the QPI modality. And for all the visible light, it's limited by about like 100-ish microns, depending on the sample, of course. For the muscle, I think only the shallow region. It's mainly because of the skin. The skin is because the whole point of having a skin is to prevent all the external, like external things to come in in our body. So it's actually very hard to image the skin. So for the muscle, probably we only image the surface of the muscle using the visible light. I have not tested a CQPI on any of this muscle or heart tissue, but it would be very interesting to look at using the CQPI because so far this is a new system. I have only imaged the organoids and the in vivo mouse brain using the CQPI.
Let's try. So we have, for example, mouse sinus node is only 100, 200 micron in thickness. So we image it all the way through. It's a fascinating topic. Let's discuss. Thank you.
Yeah.
Great.
Thank you. I will follow you up, Igor. Thank you.
Thank you.
And the next question is, can you elaborate on the machine learning, deep learning training details for the resolution improvement? Are there a prior physical knowledge used in the training? Thank you for this question. It's a very detailed question. It's a great question. So for the ACM, the artificial confocal microscopy project, The training details is that we used InfinityNet, which is basically it's basically UNet version of the network, and we the input is 3 Z's, 3 adjacent Z's. So remember, we always took 1 Z stack of the imaging samples, and then so the delta Z, you can, depending on the resolution of the modality. What we chose is 1 micron as our delta Z. So each 1 micron step, you take an image, and then you have a stack of images. And then when we put into the network, we chose 3 adjacent Z's. So Z1, Z2, Z3. Those 3 Z's were input in the network, and the network will train on those 3 Z's, and we of course iterate through this whole Z stacks. So Z1, Z2, Z3, and then Z4, Z5, Z6. So all the way through this Z stacks, we put into the network, and the network will also see the colocalized pair of the other confocal fluorescence image. And after this trained, you still do the same thing. You input 3 Z's, you will output 1 Z. So this is the key. So you input 3 Z's, the output is 1 Z. So the network is learning what's the difference between the 3 Z's, and what's the difference that in the laser scanning LIM module versus these confocal fluorescence module. So the network is trying to learn how to map from this 3 Z's to 3 Z's which have better axial resolution and sectioning. And for other details of the training details, I recommend you to read our papers. Those in the master section, we have a very detailed description of how all the details of this training session. Are there prior physical knowledge in the training? No. The network does, other than those pairs of images, the network does not have any prior physical knowledge. So the next question is, I'm wondering, actually from Jack. Hello, Jack. So Jack says, I'm wondering what is the highest frame rate? Yeah, hello. What is the highest frame rate of the CQPI? And if it is possible to use it to deduce Thomas membrane changes during action potential event? This is a great question. So highest frame rate, as I mentioned, because we have so much photons. So actually we can, the only limiting factor is the scanner. So, but the faster scanner exists. For example, we can use a polygon scanner, the resonance scanner, polygon scanner to have a faster scanning mechanism. And we will, using those scanners, for the resonance scanner, we are able to reach 30 hertz frame rate. And using the polygon scanner, we can reach about 700 hertz frame rate. So that's basically the 700 hertz frame rate is enough for voltage imaging even. That's like the fastest event that people are interested in. So for the membrane changes, I think the frame rate about 100 hertz. And also for the blood flow, if you're interested in the blood flow, maybe you want to have an image, the blood flow at 100 hertz. Yep. And it is enough. So CQPI can do all those sorts of the things. As for the Thomas membrane changes, yeah, so that's probably also like 100 hertz-ish. The difficulty is that as we see in the CQPI channel, we have all the signals coming from all the cells, right? But if we have the fluorescence introduced by the 3-photon channel, we know this cell is what we're interested in, and then we can use that as a binary map. So you can actually basically separate the information from this one cell, separate this signal from the rest of the cells that you're not interested in, and only looking at this cell, then we should be able to image some membrane changes. That's my guess. I've never tried, but as for the frame rate, it is enough because it's a linear scattering base. We have a lot of photons to image with, and also we have a faster scanner available in the market. Yes. So
yeah, that's amazing. I was just thinking, because you show so many pictures of static images, I was wondering if you just circle an ROI and plot the time series of those images, like what kind of signal we're going to be able to get. It should be very interesting because it will be I imagine a mixture of like blood flows or lymphatic or I don't know, action potentials even. Yeah.
Yeah, actually, in this supplemental of this CQPI paper, which will come out soon. So we also demonstrated the blood flow imaging. However, because of the laser, I'm using the 3-photon laser. The wrap rate is only 1 megahertz. So I did not. And also we used the Vivo scanner. So but if you use another CW laser, which you don't have, you have a bunch of photons, you don't have this wrap rate limitation, and you use a polygon scanner, and we can image very easily the blood flow. And in this CQPI paper, I did use a Vivo scanner with this the multiphoton laser, the femtosecond 1 megahertz laser, to image at the frame rate of 7 or 8 hertz of this blood flow. It's not enough, but it's also I want to mention that I imaged using 0.7 milliwatts all the way down to 800 micrometer inside of the brain. Oh, so 0.8 millimeter. So yes, so we can see the blood vessel, the blood flow all the way to the 800 microns deep, and with 0.7 milliwatts, right? So if you this is the 7 hertz, and if you're imaging about say like 100 hertz, then like 20 milliwatts-ish level of light should be enough in the 800 micron depth. For the shallow region, you don't need, So
that's amazing. Yeah.
We should try. I come here.
Yeah, definitely. Yeah, thank you so much. Great talk.
Thank you. So the next question is, have you imaged organoids over time, especially when there are multiple types of cells that are differentiating? That has been a challenge for me, but your approach might offer a nice solution. Andrew, we should talk. I have not imaged organoids over time. We just shown that this works, but of course, the reason why we developed those imaging techniques are for imaging organoids over time. And using this, we'll be able to image live organoids and also give you the computationally specificity and axial resolution that you needed. So, of course, I think we should be able to see the differentiating of the cells, and we should talk. You should reach out to me by email. And I think that's all the questions I got.
Okay.
Okay, one more. Is it possible to do wide field imaging instead of laser scanning? Yes. So actually, all the previous, like prior 2022, all the efforts in Gabby's lab at UIUC were focused on the wide field. The laser scanning we only step into the laser scanning, the confocal, since like 2020. So all we did before was wide field imaging. However, wide field imaging is poor on the axial resolution. So for organoids imaging, it's not that great. That's why we developed this ACM method to give you better axial resolution, better sectioning, and also give you the specificity of the biochemical, like the molecular signal that you want to look at. Okay.
Okay, I think we reached the limit on time, right on spot. Once more, Dr. Chen, thank you so much for your time, and to all the participants for the questions and interest. I'd just like to terminate reminding that I'm going to stop sharing this screen, I hope. Cancel
I can stop sharing.
And I would like to just share the program if I can for the next, I guess I can't. For next month's seminar will be a physical genomics trainee showcase. And in August, we have Dr. Travis Craddock from the University of Waterloo. And we'll post and invite everyone for the upcoming seminars. Thank you again, and have a wonderful weekend. See you next month. Thank you
so much. Thank you, everybody.
Thank you. Bye. Have a good day.